Well, one of the very exciting projects that we've begun at our institute is to develop a rice that is much more efficient in its photosynthesis. There are two principal ways that plants capture sunlight. And one is called C3, one is called C4, and it's based on the sophisticated chemistry and the techniques that the plants have evolved over millions of years to capture sunlight. The C4 plant is very efficient at using sunlight and using carbon dioxide, using water and using nitrogen, and typically has a much higher yield. These are sugarcane, corn, sorghum. The C3 plants are the more primitive, if you would, plants like wheat and rice, and tend to, under the same exact conditions, give lower yields than say, corn. What we are trying to do is engineer rice so that it will have the photosynthesis capacity of corn. And we expect that would increase yields of rice by 30 to 50%, decrease its nitrogen use, or rather increase its nitrogen use efficiency, increase its water use efficiency. So this could have a phenomenal impact on productivity of a given plot of land. Now in the future we know we're going to have to produce more food on less land with less water, less labor, be much more effective in our nitrogen use. So to me this Apollo project, if you would, to re-engineer the rice plant is one of the most exciting undertakings. Now, in some ways it's a very risky undertaking. Nobody's ever done this sort of thing before. On the other hand, when we look at nature, the C4 or more efficient photosynthetic process has evolved independently 40 different times in the plant kingdom. So it's something that is there and it's not that difficult. We would just like to do it in 20 years instead of a million. So that's one that. And of course the golden rice with the high beta carotene is another area that's really exciting. We have embarked on a very ambitious program to generate the genetic sequence of effectively our entire gene bank. That is, completely describe the genetic variation of all of rice on the planet, and to make sense of that data, to actually characterize the genotype, or the phenotype or the performance of the plants under a whole range of conditions, and associate that performance with the genetic variation so that we could hand a very, very powerful toolbox to breeders so they could create any kind of rice variety that could meet any conceivable set of circumstances in a way that's today not possible. We can only dream about it, but I am certain that 10, 15 years from now, breeders will be using the tools that we generate today on a regular basis.